Assalamualaikum and very good day. My name is Mazniatul Iqma and I am the head of the Department of the Research and Development at Prime Pharma. In this video, I'll be talking about our team's approach to develop a novel anti-diabetic drug. But before that, did you know that diabetes has become a major threat to our country where 1 out of 5 people are diagnosed with diabetes and the prevalence of diabetes keeps increasing year by year. Diabetes, primarily diabetes mellitus type 2, involves the body's resistance towards insulin, hence causing a spike in glucose level which impairs our normal body function. There are numerous oral treatments available for diabetes type 2, such as sulfonylurea, peglatinates, biguanides, thiazolidinediones, and alpha-glucosidase inhibitors. Among all of these treatments, insulin sensitizers such as biguanides and thiazolidinediones are the first one to be administered to diabetic patients with declining beta cell function. However, there are several adverse effects reported with the administration of insulin sensitizers, such as metformin reported to cause hypoglycemia when combined with other treatments, and thiazolidinediones, which is reported to be hepatotoxic and can cause edema, which is contraindicated to patients with heart failure. Hence, we from Prime Pharma decided to come up with a new drug that has fewer adverse effects but still potent enough to be an insulin sensitizer. In this video, I will explain one by one our approaches for drug development, which starts with target identification, then to in silico design, hit and lead selection, hit optimization, clinical trials, and drug development. First of all, for target identification, after a thorough literature review, we decided to have peroxisome proliferated activated gamma, or in short PPAR gamma, as our target protein that will bind to the novel drug. Currently, PPAR gamma is the target protein for thiazolidinediones, where it binds to PPAR gamma on the liver, muscle, and adipose tissue to promote adipogenesis and glucose uptake. PPAR gamma is also our choice for target protein as it is highly expressed in liver and muscle tissues, unlike other target proteins that are not specific to those tissues. Previous research also discovers numerous PPAR gamma agonists such as amorphotin B, monasin, and ferritin that can act as insulin sensitizers. After knowing our target, we will retrieve the protein 3D structure from protein data bank to be used for in silico design. During in silico design, we will do virtual screening to virtually screen the properties of the compounds and their interactions with PPAR gamma. During this stage, our team will be divided into several groups with each doing different virtual screening approaches. One team will do similarity search to compare the structure of the compound to thiazolidinediones as an established PPAR agonist. Another team will be tasked to do pharmacophore modeling of PPAR agonists to identify the core structure that are responsible for the insulin sensitivity properties. Besides, another team will be focused on QSAR modeling to further understand the insulin sensitivity properties of all of the compounds. And another team will be focused on molecular docking and molecular dynamics to know and compare the binding affinity of the compounds to PPAR gamma. After that, for hit and lead selection, all of the in silico data will be compiled together and be evaluated. Then we will choose 50 hit compounds that have the highest binding activity and have the highest similarities to thiazolidinediones and also have common pharmacophore. Then our cell culture team will perform in vitro high throughput screening onto those 50 hit compounds to screen for compounds with the highest potency and then out of the 50 compounds, we will only choose the best 3 compounds as lead compounds. Then we will proceed with lead optimization, which involve both in vitro and in vivo studies. Our team will start by doing in vitro assays like MTT assay to evaluate the cytotoxicity of the 3 lead compounds. Then our animal study team will proceed with doing in vivo tests to determine the pharmacokinetic and bioavailability of the lead compounds, and this will be done by administering the compounds to mice at different route of administration and also different concentration. Then acute and chronic toxicity test will also be done by extrapolating the cytotoxicity data from the in vitro assay. Only then, one compound will be chosen for the next stage, which is clinical trial on humans. For clinical trials, we will collaborate with the local clinical research center or CRC to recruit volunteers. Clinical trials will involve 4 phases, which first be done onto healthy humans to evaluate the compound safety. Then the second phase will be done to evaluate potency, and then the third phase will be done to evaluate both safety and efficacy. If the compound passes the 3 phases of the clinical trials, we will submit application to the FDA for a new chemical entity. Then, upon FDA approval, we will continue with phase 4 trial, which involve the development of the approved compound or drug. Then, we will analyze the clinical trial data to evaluate the drug safety and efficacy, and We will alter the structure of the drug if any of these two parameters are off. If any of the stages from this strategy does not work, we will repeat the process again from where the problem started to rise until we get a new drug with high efficacy and safety and also few side effects. That's all from our company. I hope this information helps. Thank you for listening.